Hello, everybody. Welcome to Club EvMed. I guess I should say I looked it up because I got it wrong last time. Club EvMed Virtual Evolutionary Medicine Conversations. We like it to be a conversation. We like a presentation, but we also like to hear a lot of questions, a lot of perspectives. We'll give things just a few more minutes. And while we're doing that, I will thank Jennifer for all of her organizational help and keeping these going. And also thank our committee that helps decide which talks to focus on and keeps a nice diverse portfolio of speakers coming through. Stay tuned for our next set of speakers. I don't know how much of that is up on the Club EvMed website, but we actually really need to get that list up, at least of the speakers, even if not the topics. Okay, great to see so many people tuning in. And thanks to those who have their cameras on. Always nice to see some faces. And feel free to turn that off when you... Oh, hey, we have a hand from Bill Pan. Nice to see you, Bill. Okay. I think probably have enough people to get started out too. As soon as I say that, more people start coming into the waiting room. Well, I'm just going to go ahead and get started. I'm Charlie Nunn. I'm from Duke University, and I run TRISEM, the Triangle Center for Evolutionary Medicine. And TRISEM is the organizer of Club EvMeds, and Club EvMed, we like to call it Virtual Evolutionary Medicine Conversations, Club EvMed Conversations, because while we want to share findings and get everyone together, we really want to hear from people and try to build the community in evolutionary medicine. Club EvMeds are organized by the International Society for Evolution Medicine and Public Health. I just posted that into the chat, as well as our journal, the Society's Journal. We're going to hear just a little bit about that in a few minutes from Vaughn Cooper, the editor-in-chief. Please renew your membership to the Society. If you're not up to date, please do that. And please consider submitting articles to the journal. Now, Club EvMed is also organized by a bunch of different evolutionary medicine centers, and so I'll paste that in next. I think there are about 10 or 12 of them, and hopefully I have them all here from around the world. And we see many people from these different centers on the call today, so thank you very much for joining. Okay, I think that's all I have to say. I'll turn things over to Vaughn to say a few words about the journal, because this is a Club EvMed where we are highlighting one of the journal articles. We try to do this once or twice a year. It's one reason why you might want to submit to the journal as a way to maybe be one of the highlighted articles and really get your work a little more attention through Club EvMed. And I'm sure Vaughn has a few other reasons why you should submit your articles to the journal.
Well, geez, Charlie, I think you've pretty much already given the great arguments for why it's great to consider EMPH as a journal. So, of course, it's our official society journal. Our reviewers are often from us or from people who we wish to engage in society, and I've been delighted in my first sort of year as editor-in-chief to see how many authors were happy with the constructive and supportive review process. We do attract terrific papers like the one you'll be hearing about today, and I'm happy to say there are more in the queue. So if you do submit your good relevant work to us, you won't find it alone. And then we have a really great history of sort of feature issues, which are sort of thematic issues. One that's going on right now that's enjoying great success is basically evo dynamics in cancer. So you're already starting to see some of those articles coming out in the journal, and several more in the pipeline. And then next year, I'm delighted to say we have an exciting issue around sort of ongoing evolution and pathogens during infections planned. I'm not going to say all those details until it's official, but you'll see notes soon. So with all that, I think EMPH is continuing its strength, as having an excellent year, and we hope you will consider submitting your work there, and please share the word.
Okay, thank you, Vaughn. And yeah, get on the table of contents if you're not on the listserv for that. And there's also the Williams Prize, so the best article every year is eligible. Well, all articles are eligible for the Williams Prize, which goes to the best article in the journal. $1,000, is that right? I think it's $1,000 for the Williams Prize. Okay, and now I'm going to turn things over to Bernie Crespi, who's going to introduce our three speakers today, and they will talk about the article that they published in EMPH. Go ahead, Bernie.
Thanks, Charlie. It's my pleasure to introduce our three speakers. They include Katya Coel, who did her PhD at the University of Michigan, is now an associate professor at Emory University.
I'm actually a full professor, but thank you.
Did I pronounce your name wrong?
It's Cole, but that's fine.
Cole, Dr. Cole. Set the record straight. Thank you. Let's see how I do on Rustam's name. Rustam Antia.
Yep. Good
No
luck for
corrections.
government
Excellent.
work.
Yeah, that's right. And Dr. Antia did his degree at UMass Amherst. He's now a professor at Emory. And Dr. Jim Bull did his degree at University of Utah. And he's at the University of Texas at Austin and also now at the University of Idaho at Moscow. And Rustam, are you starting us off here?
Sure. So, oops, now it's telling me I have to do something about share. Share. Oh, sorry, it's asking me to share settings as usual. I'm going to have to, let's see, Zoom may not be able to record the screen later. Okay. Whatever. Let me try sharing again. Yep, it should let it do it.
Okay.
Okay. So thanks for inviting.
Rustam, do you want to go to full screen maybe?
Yep.
Yeah. Okay. There we go. Looks perfect.
Uh,
Except for another. Okay.
yeah.
Thank you.
So thanks for inviting us to talk about this paper. Please ask questions as the talk progresses. Jim and Katya don't have to be told to interrupt. They're very good at it, and that's really helpful. And I encourage anyone who has questions not to wait for the end, but to ask them as it goes on. So we've been sort of interested in waning immunity for a long time, and respiratory viruses as well. And so one of the sort of big questions in the field can be put very simply as, why does influenza exhibit antigenic escape, but measles doesn't? Okay, let's see. Okay, so I'll start by, do I have to minimize that? Is that better? Yeah.
Looks okay.
Yeah. So we can start by sort of looking at some of the ideas of why influenza exhibits antigenic escape, but measles doesn't. And one of them is simply to look at the protein to which you make an response. And most people, when they talk about immunity, talk about antibody responses. And if you look at antibody responses to influenza, they're made to a protein which is on the left, hemagglutinin. And you can see that that protein is very tolerant of having mutations. And lovely work from Jesse Bloom's lab has shown this sort of in this figure on the left. You see a lot of places are colored red. Those are the sites to which antibodies bind. They can vary. And that's not so much the case for measles. So that's one hypothesis. Another hypothesis, also from Jesse, is the immunodominance hypothesis. If a virus has lots of codominant epitopes, there are lots of targets in this scheme for the antibodies to bind to and neutralize the virus. So that sort of a virus finds it hard to escape from immunity. While if there's a single or just a few immunodominant epitopes and the virus can mutate it, it can escape from immunity. It's not that clear that this is actually a mechanism operating in influenza, but it's an interesting mechanism nonetheless. After mutational tolerance and immunodominance, there's the generation of antigenic escape can be facilitated by having intermediate levels of immunity. And in a wonderful paper, Brian Grenfell and his colleagues back now in 2004 once sort of pointed out that the strength of immune pressure, when the strength of immune pressure is sort of at an intermediate level, the virus adaptation rate is greatest. And they have this lovely cartoon which sort of illustrates it. When there's high immune pressure, there's low virus abundance, but there's strong selection. And again, when there's low immune pressure, there might be high virus abundance, but there's low selection. And so the sort of Goldilocks principle holds. At an intermediate level of immune pressure, you get quite a lot of virus and you have some selection. And in their paper, they sort of focused on the generation of variants within a host. You can see that in the panels on the right. In naive hosts, you have lots of virus, but very little selection pressure. So on the panel below, there's relatively little selection for immune escape. At high levels of immunity, there's almost no virus, there's almost no immune escape. And the Goldilocks principle, works at intermediate levels of immune selection. Okay? And so what we did, and I should mention here, there's a nice paper which we only got to know about more recently by Bajpai and Watve in Krios in 2023, and they have a similar, they suggested this too, is waning immunity drives the spread of antigenic variants. And it's really this intermediate level is there for not just the generation of variants, but also the spread in the population. So the conventional view, sort of illustrated in the left, is that waning immunity is one thing, immune escape is generated separately. Both are required to facilitate reinfection. The alternate view, which Bajpai and Watve mentioned and we worked on in this paper, was that waning immunity drives immune escape. And so that's the route at which it allows for reinfection. Okay. So to sort of make a figure for this, a sort of schematic, one can look at how fast the virus grows within the host as a function of time after the host has been infected and recovered. So shortly after infection, there's lots of immunity. There's a high level of immunity. The wild-type virus grows actually has a negative growth rate. That's sterilizing immunity. If you get exposed to the virus doesn't grow in you, it gets controlled. If you have low amounts of immunity, the virus can grow. The mutant does somewhat better than the wild type in the presence of immunity, but has a cost in the absence of immunity a long time after infection. And sort of just to mention, this cost is needed because otherwise the mutant would always do better than the wild type if there was any level of immunity in the population. So rather than make a complex model, we made the simplest model we could think of to explore the conditions under which waning immunity would provide a selection pressure for escape variants to spread at the population level. So in a classic SIR model, we had susceptible hosts S, they get infected by the wild type virus and become I subscript W. They recover and become immune. And then this immunity can gradually be lost. When immunity is lost into the partial level of immunity, the wild type can't grow, but the mutant can grow a little bit, as indicated by the parameter epsilon, which says it can grow only somewhat. And then partial immune hosts can lose their immunity and go back to susceptible, where they can be infected again by the wild type as well. Now, the mutant can have a slight cost, so it can't. The mutant can infect both susceptible and wild type, but with some slight cost. And this slight cost is there, as I said, because otherwise the mutant would just invade all the time with any level of immunity. It turned out that this system of equations was simple enough to be analytically tractable, to allow us to look at how the different parameters would affect the allow the invasion of a mutant. So the way we did that was very simple. We just considered the solid arrows, which are at the bottom, to look at what would happen with the wild type virus when it reached equilibrium. And we can get a steady state for the susceptibles, the wild type virus, the immunes, and the partially immunes at equilibrium. And then we can look at whether the mutant would invade that equilibrium. And you can do that, and you can solve, and you can get an expression for the reproductive number of the mutant. And it turns out to be nice and simple. The reproductive number of the mutant depends on the cost of the mutant. The higher the cost of the mutant, the lower it would be. And it depends critically on p hat, which is how many partially immune hosts there are at the steady state with just the wild type virus. So if we want to look at this, we can make a simple plot. And focus on the left to begin with. The panel right to the left tells us the number of partially immune hosts as a function of the R0 of the wild type virus, which is on the y-axis, and the immunity, how long immunity to the wild type virus lasts. So in other words, how immunity wanes on the x-axis. And you can see that, as you might expect, as the reproductive number of the wild type virus increases, you get more partially immune. And the largest number of partially immune happens when the waning rate is intermediate. If the waning rate is really fast, everyone loses immunity right away and becomes susceptible. If the waning rate is very slow, people stay in the immune compartment and never drop to being partially immune. So you can see in the panel just to the right of it, where the number of partially immune individuals is high, the invasion potential, which is the lighter colors being invasion, is higher. And there's also a region in which invasion might not happen, right? If the R0 of the wild type was sufficiently low and the cost of immunity is sufficiently, the cost to the antigenic variant initially is sufficiently high. So that's what we see. It behaves like we expect. If we increase the cost to having the mutant, then the invasion range decreases. If we make it so that the level of partial immunity provides more protection against the mutant, again, the level of the range for invasion decreases. So what we have over here is intermediate ranges of waning, which is on the x-axis, show the allow the facilitation of new variants. So that's really a very simple idea, and sort of the range at which it happens is is captured in the model. Now, you might think that really long level immunity is held by something like measles, while shorter level immunity is due to influenza. And so we are back to this question, what about measles? So one would think that measles is a respiratory virus. We get it by the respiratory route. Why doesn't measles also exhibit strain variation? And really the answer to this was beautifully described by John Udell in a lovely paper back in 2021, where he talks about the duration of immunity to different viruses and how it depends on their route of transmission, and critically not just their route of transmission, because both measles and influenza get transmitted by a respiratory route, but its site of replication. And so over here, this is a figure from his paper. He has different viruses on the left, the ones which are coded in yellow, which include measles over here and influenza. The initial site of infection is the airway, but interestingly, measles replicates in the airway and doesn't require systemic infection for replication and dissemination. In contrast, measles, mumps, rubella all require systemic replication. And what John Udell pointed out was that if you have systemic replication, you have durable immunity, you have wonderful vaccines, and as we sort of describe over here, you're not likely to get selection for variants at the epidemiological level. In contrast, if you don't have systemic immunity, immunity in the respiratory tract where the virus is replicating is short-lived, you don't get durable immunity. So in this view, mutational tolerance is not a cause of immune escape, but really might in large part be a consequence of immune escape. The hemagglutinin molecule has sort of acquires mutations and then over time gets selected for mutational tolerance. So I'll end by just mentioning a few other considerations, one of which is really understanding what determines the rate of waning of immunity. In particular, how does immunity wane in the respiratory tract? John Udell and others have done a lot of work on this, but there's still lots of unanswered questions. And another consequence of waning immunity and the generation of escape is that it might limit the regime under which we can get and generate universal vaccines against some respiratory pathogens like influenza, which replicate in the respiratory tract. I'd also like to highlight a few papers which people would find sort of useful when thinking about this. A classic one is a paper by Wallace in the late 1980s in the context of HIV, where he asked whether lack of immunity, low levels of immunity, could form stepping stones for the evolution of viruses. Another interesting set of papers is by Julia Gorgon-Grenfell, as well as Katya and her colleagues, which look at the complex dynamics of multi-strain pathogens. Neil Ferguson and Alison Galvani and colleagues have looked at sort of short level and long level immunity to influenza in a very nice paper in Nature in 2003, and sort of looked at the phylogenetic patterns that emerge from this. And as I said, Bajpai and Rottwem recently have mentioned how waning immunity drives can drive the evolution of variants in the context of SARS-CoV-2. So with that, I'll stop over here and hopefully take some questions.
Great job, Rustin. That was really nice. Really nicely explained. I mean, I'll just start with a quick question. I saw Bernie's hand go up right as I was going. I wonder if you could say more about the costs and what these costs are, because it occurred to me that one cost could be the cost to the host, right? If you kill the host or make the host so sick that further transmission isn't possible, that could be a kind of cost. And if that's one of the costs to the virus, that gets into the evolution of virulence and things like that. So I'm just wondering, what kind of costs are you thinking about, and have you thought about, how this might extend out to some other questions within disease ecology and evolution?
Yes, the evolution of virulence is a fascinating question, particularly when the virulence kills the host and the host happens to be humans. They unsurprisingly get very intrigued by it. But as sort of, at least some of us who've sort of contributed to writing a lot in the field, find there's less and less that we've written that actually has interesting consequences. There are too many caveats to it. So for example, for influenza, there's very little selection for killing the host, even though you're absolutely right. If it was severe enough to kill the host, there would be no transmission, so there'd be a high cost. So that could be one cost. But what we are more thinking about is simply just that the virus is a little less fit.
A little less fit. I mean, if the person had really, if they were very sick and removed themselves from their social networks, that's another way that it could be a cost to the virus.
And Andreas Handel has done some work on that. He pointed out in some early work I did with Bruce and Bob May, sort of the optimal level of virulence is where you almost kill the host. And Andreas pointed out almost nothing evolves to there. And one reason might be is people stop bumping into each other a lot before they feel that sick.
So if I can just kind of add on to that a little bit, in terms of the cost. So there have been some nice instances kind of in flu actually indicating that some of these new antigenic variants actually do have fitness costs in terms of replicative fitness, and so one example of that is in a Cell Reports in 2015 by Chambers et al. Looking at the Texas 12 strain, there's kind of this mutation so for flu, new antigenic variants really come about from these small number of residues kind of on that hemagglutinin head, right? So it's really kind of where this receptor binding domain is, right? And so they kind of documented that this new antigenic variant actually has a fitness cost that's been established also in cell culture, where if you kind of have a comparison of kind of how the viral just the amount of virus kind of in a dish in the same set of kind of cells, the wild type versus the one that has this antigenic antigenicity impacting substitution, the one that has a substitution grows less rapidly, right? And that's because it has lower cell binding avidity. So that's been pretty well established, but then in the context of actually antibodies against kind of the wild type one, it grows better, right? And so really kind of the cost is more like replicative fitness cost in terms of which translates to then viral load, right? Or viral titers, which then influence onward transmission potential.
That's interesting.
I had a question about the public health implications. There's a number of ways to get partial immunity, and one of them is waning, and the other is imperfect or not totally effective vaccines. And this is certainly the types of vaccines that we have for we have for COVID. So the question then becomes, how do you think this might apply to the idea that imperfect vaccines can be driving the evolution of the viral agent, perhaps not just in this context of waning immunity, but also in terms of applying a very specific kind of, monovalent selective pressure. So the idea is how we're getting, in principle, we're getting selection from both, and they could go in the same direction. I don't know who I'm aiming that question at. I guess all three of you. It's a hard question, probably impossible.
Well, I'll take a stab at it. Yes, immunity. So what you're suggesting quite correctly is that if vaccination leads to intermediate levels of immunity, it would drive up the probability of immune escape. Is that correct?
Yes, so that's one of the main
So
ideas.
it's a fascinating one. I haven't thought about it much, but one might think that there are two approaches here. One of which you might not want to vaccinate everyone, but focus it on those most at risk to minimize the chance of escape. And the other one is you might want to vaccinate frequently enough that you're actually taking people out of the intermediate level of immunity and moving them into high immunity. So yes, in principle, it could happen, and it might, and I haven't thought about it carefully, but it might depend on how you tailor your vaccine program.
Rustin,
Right,
do you want to mention that measles paper you showed us the other day that it looks like?
Go ahead, Jim.
So Rustin found this paper on antibodies against measles, and basically for people my age and maybe considerably younger that actually got measles, our antibody levels are very high. Starting and whenever the measles vaccine came in widely, you look at people from that class and their antibody levels are extremely low. So the question you raised, Bernard, I think is very relevant in that case, because you're going to have increasing population with very low levels, low levels of immunity to measles, and we don't know what the consequences of that are.
And I think just to chime in kind of actually for flu, it's a globally circulating, these flu viruses are globally circulating, and so I think it depends not only, what vaccination rates are kind of, for example, here in the US, but actually, what they are kind of globally, right? And then of course, kind of what vaccine effectiveness really is, so there have been a few papers that have been published looking at, can vaccination actually drive the evolution of antigenic variation in flu, right? And I think kind of it seems like there's not very strong kind of evidence, kind of indicating theoretical evidence, kind of indicating that would actually happen at the rates at which vaccination happens, and kind of at a global scale, and the current levels of kind of effectiveness that's estimated. But I think for something like measles, where vaccination rates are much higher, and then of course kind of in places where vaccination is lower, there's also natural infection, right, which is creating kind of immunity. I think it can be somewhat different. And I think the question, also, is very interesting for things like, varicella zoster, where, chicken pox, right? And resurgence of kind of shingles also maybe, in terms of, patterns of kind of natural circulation of chicken pox versus kind of standing immunity, and if that gets boosted or not.
Right. I mean, it really seems like both a mathematical question where the math is required, but also kind of a moral one, because you would have the short-term advantages to individuals from vaccinating them, but at least in principle, those short-term advantages would necessarily come with the promotion of the long-term evolution of the antigenic variants and the continual generation of strains that were better able to infect people. So maybe someone wants to model that one. I don't know. It
I,
seems important.
yeah, I mean, Bernie, if I could jump in, although I was going to ask a different question, this notion that vaccines cause variants is, deeply problematic. And I know several of you have written about it. I wrote about it, and that's just, I mean, it's wrong. I don't think that there's good evidence for it, and I think rather the major point of this paper is the role of waning immunity or inadequate levels of vaccination. But the experts are here, so please set me straight.
Why is it
Just to
wrong?
just to kind of add on to that, I totally agree, Yvonne, that there's no, compelling empirical evidence that shows that, vaccination has really kind of sped up, antigenic evolution in viruses, and then we have to think also, in terms of, SARS-CoV-2, if we think about kind of, the new antigenic variants for SARS-CoV-2 that, that emerge, like Alpha and Omicron, of course, those, very likely came from chronic infections, right? And so, to decrease chronic infections, you're also thinking about kind of reducing the levels of circulation. And one way to do that is through vaccination, right? So that's kind of another argument. And in that case, that if we're thinking about pathogens where, chronic infections, who knows whether that's going to be the case in the future for SARS-CoV-2, right? But at least kind of, early on, in the first, few years of the pandemic, increasing vaccination lowers the number of people who are infected. It of course kind of lowers disease more so. It's more, vaccine effectiveness is higher against disease than it is against infection, but doesn't decrease infection levels as well, right? And then that's going to decrease the number of people who are also, to the extent, that, immunocompromised and immunosuppressed, where maybe direct vaccination of those individuals won't actually protect them against infection as much, so that kind of herd immunity would actually, through that kind of causal chain, also kind of slow down actually antigenic evolution if you're thinking about it coming from chronic infections.
Bernie, did you want to say anything more about that? I think you had started to say something.
I mean, is there empirical evidence relevant to the question of vaccination, especially imperfect vaccination, driving evolution? I mean, it seems like a selective pressure to me. I can't speak to how strong a selective pressure it would be, but is this a question of absence of evidence or evidence of
So if you look actually at there's several kind of within host studies of acute infections in humans, for people. So this is Dennis et al. It's out of Tom Friedrich's group, and then some work also of Adam Loring's, where in his group, so where you kind of stratify by whether an individual was vaccinated or not, right? And you see whether, is there more viral genetic diversity, in a breakthrough infection, right? In individuals who are vaccinated that season versus not, right? Do you see more antigenic variation kind of arise in individuals who were vaccinated that season, but still, had this breakthrough infection? You don't see any evidence for that at all. You see new antigenic variants come about in, again, kind of prolonged infections, in individuals that just can't clear kind of an acute infection because of some underlying conditions or treatments.
Would anyone else like to weigh in on
Talking
this?
too
Really
much.
interesting set of questions. Anyone else in the audience who would like to weigh in or ask related questions before we turn back to Vaughn for regional question?
I am working on this topic. I sorry to just jump in. I'd
Please.
like to ask Vaughn why he feels so strongly about it not driving the evolution of new variants.
Sure. I mean, I think I'm basically summarizing the arguments that Katja just made about the fact that that vaccination does not increase the generation of new variations. However, in an in a population sense, right, it does create sort of fertile ground for variants that may exist that that would not ordinarily find a home, right? So at a population level, eventually you wind up getting selection for escape, but you would get that level of selection for escape anyway due to naturally occurring immunity. And I think the real question is, does the immunity generated by vaccination versus naturally occurring immunity substantively differ to accelerate or decelerate the rate of the evolution of escape variants? And again, I defer to Rustam and Jim and Katja here. I'm a little bit better than armchair.
Okay, thanks.
Okay, other questions along this line? Yeah, we do. Alex, I don't know if you want to ask your question directly, or I can read it off we can all read it off the chat.
Yeah,
And also Bill, I see Bill. Go ahead, Alex.
yeah, I can just read it. Yeah, so I guess I mean, thanks for the presentation. First of all, it was really interesting. I guess, yeah, normally when I've thought about incomplete vaccines, I've thought about situations with like pneumococcus, where there's all these different strains circulating around and the vaccine only contains some of the serotypes, and then obviously select those ones out and the rest take over. And then I guess the scenario you're presenting is kind of a different one, which feels kind of like competitive release. I don't know if that's the right term, but yeah, I guess how important is that distinction for these questions?
Sure, it's a wonderful, thanks for raising that point. It is a very important distinction, and Mark Lipsitch and people have done some very nice work on sort of with strep pneumonia. If you vaccinate, you've got a wide range of strains which are competing with each other. You vaccinate in this is to take out the ones which are most dangerous, because you're likely to get the others expanding to fill the ecological niche. And again, more recently, there's a nice paper by Jess Metcalf and colleagues, which talk about sort of ecological space and how it gets filled by pathogens. I think it's in Trends in Immunology, or one of the Trends journals.
I can also cite a paper, I think, by Rustam and Veronica Zernitsnia about kind of maybe you can talk about it a little bit more, because I think it's relevant here also, right?
Oh, well, it's you can make a just so story, which is that if you have a bunch of strains in the population sort of seeding a seasonal influenza outbreak, and you vaccinate against one of the strains early, even if if that strain was going to have dominated, by taking it out, you're going to just give ecological space to the other strains to come in, potentially. And so the role of that is sort of not known. There's we looked for evidence sort of which would support it. We didn't find any that went one way or the other.
I feel like I'm going to need to rewatch this and write down all these citations. I mean, there's so many great papers everybody's bringing up. Like, have a whole graduate seminar on these topics. Anyhow, maybe we can ask the AI to do it.
I saw a note in the chat by Bill Penn saying that there's evidence that there's incomplete polio vaccination. There's evidence for that driving different oral polio strains and mutations. Bill, can you talk a little bit more about that if you're still there and willing to?
Sure. I have to put my phone down because I'm about to leave. So that has to do with the vaccine-derived circulating polio, where if you are giving people vaccines, and it might actually this might actually be something Vaughn was referring to, but if you're giving people certain injectable vaccines, you're usually protecting them against OPV strains 1 and 2. But then when those get into circulation, because you're defecating it out into open sewage and things like that, if people are unvaccinated, you can contribute to how those strains are taken up by people who are not vaccinated, and they get different types of mutations of OPV1 and 2 that are affecting them. And then, of course, you've got the oral polio vaccine, which is mostly against OPV3. And, that's a live attenuated virus versus, the injectables are, I'm sorry, those are, the OPV3 is a live attenuated virus, and OPV1 and 2 vaccines, which is what we get in the United States, those are against all three, but it's not as effective against OPV3 as it is for the OPV1 and 2, which is why you can get these circulating vaccine-derived polio outbreaks. But the point is, there are studies that demonstrate that if you have incomplete vaccination, you can drive certain types of polio strains up in communities when you get incomplete vaccinated communities. So just saying that some of that evidence does exist.
Thanks. It's really helpful.
Okay, back to Vaughn for your original question you were going to ask.
Yeah,
Or
yeah,
something-
and I just put part of it in the chat. Thanks, but I was struck by that table that you showed, Rustam, from UDL. So I pulled up the paper and noticed it's pretty short, but it's a nice summary. And then I noticed it's been well cited, but mostly cited in the context of SARS-CoV-2. And I'm wondering then how broadly this is being considered among, other targets for vaccinology, and also whether it's been tested. I mean, I don't know how you'd test it experimentally, but you could imagine a virus that maybe has been engineered with different tropism in an animal model, and then you could basically test vaccination against the virus that has limited circulation or limited spread versus more systemic spread, and then track durability of humoral immunity. And then that led me to your paper, and I guess I invite you to comment on if you wish.
Well, I'm not sure which paper you're talking about.
You have yeah, it's in the chat. This Saha et al. In medRxiv.
Yeah, so Ananya was a graduate student who worked with Katya and myself, and perhaps the nicest data on waning immunity, well, almost certainly is from Mark Slivka's group. I'm not sure how many people are sort of familiar with this. The main problem is that grants last 5 years. Waning immunities typically, apart from influenza and a few other pathogens, go slower than that. So the question is, how do you measure something given that you'll want to write a grant for longer than they'll give you the money for? And that was sort of solved brilliantly by Mark Slivka, who was at the OHSU. And what he did was he noticed that people had, in the primate center, had collected blood samples to look for cross-species transmission events and potential problems with those and just stored them for a period of 30 or so years. And they'd filled up their freezers with them, and fortunately, not just for the people, but also for science, they weren't being used for anything because there weren't any of these cross-species transmissions. And what Mark did was he did a heroic sort of study where he measured the rate of waning of antibodies to a variety of virus and vaccine antigens in these people over a period of 30 years. And the lesson of that paper is Mark did the first analysis quite well. We did a little bit of additional analysis, but we then realized that this waning, people sort of think of waning at a constant rate, and it's not at a constant rate. It's rapid initially and slower later. And really, Peter Tunis and a few other people have pointed out it's a long tail distribution, and it sort of looks like a power law, at least for government work. And that's systemic immunity. That's not respiratory immunity.
Thanks for the summary. I guess if I could circle back to the other little bit of the thread is, have there been further studies or dialogue about this model about respiratory versus humoral immunity? In other words, is this accepted?
Yeah.
Yeah.
It's hard to measure respiratory immunity very well, right?
Right. Exactly.
It's easy to get a small blood sample, but people don't like being sampled invasively in their—
Yeah, lavage is no fun.
Yeah.
Yeah. Thanks.
Okay, we have, I think we'll make this the last, major question, then I'll just close with one kind of maybe fun question. And I don't know, Parejoan, I don't know how to pronounce your name. I'm sorry if I've got that wrong. Would you like to ask your question?
No, it's regarding thank you very much. It's regarding the residual immunity. I've just attached 2 papers regarding the pandemics, influenza pandemics from 2009, in which all the people were protected because they had previous immunity. They say from the 1918 pandemics, but another paper says pre-50s H1N1 conferred this protection against the swine. So then the waning of immunity in this case is not. What do you think about that? Sorry, but
Any thoughts, Rustam or Katya? Jim, on this?
Sure, you're right. But sort of 2 bits over here. One bit, to look at the waning of immunity to the 1918 sort of, I thought I understood it until Lona Simonson and Cecile Riboud sort of pointed out that there are lots of puzzles that people don't answer. Maybe we should get them to write a paper on the puzzles, because people only write papers when they think they have answers.
that's true.
I think, and with respect to these papers, I mean, I think I don't. I'd like to look at these 2 papers, so I'm glad you sent the citations. I think a lot of these studies also indicate that there's protection, long-term protection against kind of more disease, right? Or symptom development rather than infection. So that's one issue. And
well.
And then there have been studies, by, Adam Kotarski, for example. He's got a nice paper, showing kind of how frequently individuals get reinfected with flu, and it's on the order of about 3 times a decade. So that's. So I mean, clearly there's an interaction between waning immunity and antigenic evolution, right? It's antigenic evolution can drive further reinfection, right? So, and then of course original antigenics, and that all has to be kind of, there's a lot of research going on, of course, kind of looking at kind of history of infection and how much immunity there is to varying different strains, right? Yeah. I don't know if Rustam, you want to add anything?
But, well, Rustam and I at least have talked about this because Jesse Bloom had a similar paper pointing out that it didn't appear that older people got reinfected with the H1N1. And so we've thought about modifying our model to ask what happens if, you have very specific long-term strain-specific immunity, but you have waning immunity to things that are some distance out. So yes, thanks for those additional references. We've thought about this. We haven't pursued it yet. And I think this is a good time to emphasize our model is really simple. We deliberately left out lots of things like cross immunity between the wild type variant and the mutant and things like that. And we think, there's probably a small chance any of it's right, very right, but there's just a lot buried here, and there's so much we don't understand. So we aren't pretending we've solved anything here.
Okay, we're almost at the top of the hour, and I just wanted to close things out with a kind of general question for the three of you. I mean, it seems like you guys have really good energy, really good synergy together, working together. I'm just wondering, what is the genesis? What's the story behind this paper? How did you, end up working on this, working on it together? I just would love to hear the story. And if you have anything you're working on next, is there an extension of this you envision, or some kind of empirical test, or I don't know, phylogenetic, Katya? We haven't heard anything about that. So just love to hear that story, and then we'll close out.
Rustam, you want to tell a story since?
Yeah, I was going to say it's all Rustam's fault.
No,
So
yeah, it's my fault, but Jim did all the work. We had to make sure older people sort of do something. So I was in the sort of Goldilocks zone. The young person, Katya, and the old person, Jim, did the work, and I just sat in the middle. As a former graduate student said, he did all the work, I did the talking.
Yeah, and Rustam's actually right next door. We decided not to be on the same computer just to make it easier. So there's a lot of conversations. And it's been a really nice opportunity to work with Jim also, who I met, I think, when I was an assistant professor. Now that I'm a full professor. No, I'm just joking. Yeah, I think I met you, Jim, I think many, decades ago, so it's been great kind of catching up again. In terms of actually next steps, one thing that, in terms of, Charlie, you mentioned phylogenetics. We're starting to actually look at kind of, these certain kind of, for flu receptor binding domain kind of substitutions, that are known to actually be responsible for these major antigenic changes in flu within H3N2, and seeing whether like certain approaches that have been really kind of elegant approaches that have been developed in other people's groups, some Derek Smith's groups and also John Barton's groups, whether some of those methods are actually kind of applicable, so we can actually see whether we can find evidence for, like basically test the hypothesis, do we see a cost initially, and then do we see kind of a net, net advantage of these antigenic mutations kind of as, kind of the wild type virus kind of establishes. And so really kind of empirically kind of test it a little bit more with a huge amount of sequence data that's available for H3N2.
Okay, you might have noticed I got dropped off the call after I asked the question. I don't know, everything froze up for me. So I'll have to watch the recording to hear the whole story there. But thank you. I hope everybody had a chance to answer. I'm not sure if you all did. But we should close down because we're at the top of the hour. Thanks everybody for tuning in. Sign up for our listserv if you want to make sure you know which one of the Club EvMed, what Club EvMeds are coming up. And hope to see you at the next one. Thanks everybody. And thanks especially to our speakers, and to Bernie and to Vaughn.
Thank you.
And Jennifer. Thanks, everybody.